Welcome to the presentation for Chapter 7, Section 1, Life is Cellular. Please make sure that you are filling in the notebook pages for your interactive science notebook as we go along. Make sure that you are pausing the video and getting down every piece of information so you can get full points. Okay, the learning objectives for this lesson are as follows. Explain what the cell theory is. Describe how researchers explore the living cell, and distinguish between eukaryotes and prokaryotes. Okay, so to start this lesson, we're going to be talking about microscopes. The very early microscopes were developed in 1665, and the scientist named Robert Hooke used an early compound microscope to look at a thin slice of cork, which is a plant material. The cork looked like thousands of tiny empty chambers, but he was able to see it very clearly, and he was the first scientist to call these individual chambers cells. And what we know a cell is that they are the basic unit of life. Here's kind of what his drawing looked like. You can see that there's a couple different materials there, and they all look pretty similar. The one on the left labeled B is very uniform and everything is in straight lines, and then the one on the right is a little more circular. So then in 1838, another scientist named Matthias Schleiden concluded that all plants were made of cells, not just the corks that Robert Hooke was looking at. The next year, in 1839, Theodor Schwann stated that all animals were made of cells, not just plants, but animals too were all made of cells. And then a little bit later in 1855, Rudolf Virchow concluded that new cells were created only from the division of existing cells. They couldn't just be put together out of nothing, they had to be created by these existing cells. And these three discoveries, along with Robert Hooke's original discovery, these led to what is called the cell theory. So your first key concept question asks, what is the cell theory? Well, the cell theory states that all living things are composed of cells. Cells are the basic units of structure and function in these living things, and that new cells are produced from existing cells. So those three things that those scientists concluded along the way are what our cell theory states. So the microscope that Robert Hooke looked at originally, not to mention the other three scientists in the 1800s, they were pretty basic microscopes. And over the years, technology has advanced just like everything else, and they've come up with this wonderful, huge microscope called an electron microscope. And this is able to reveal details that are 1,000 times smaller than those visible in the light microscopes. So the light microscope is going to just be like the same type of microscope you would have used in an elementary or middle school science class to look at cells on slides. This electron microscope can be used to visualize only non-living preserved cells and tissues. So only things that are no longer living and that they've been preserved and they are on the slides that go in nicely. So nothing living can be looked at in these electron microscopes. Then there is another more specific type of microscope called a transmission electron microscope. This is also termed a TEM. These are used to study cell structures and large protein molecules. There's a couple examples here. The specimens that they use must be cut into ultra thin slices, so very, very, very thin in order to see the structures in these types of cells. Another type of electron microscope is called a scanning electron microscope. And you can see just by looking at the pictures between this slide and this slide that there is a huge difference between what can be seen. So in these scanning electron microscopes, or SEMs, they can produce three-dimensional images of cells. So it's a more of a lifelike, realistic image. And also the specimens do not have to be cut into slices. They can just be placed whole as long as they are still non-living and preserved, because it is still an electron microscope. Okay, here's another picture of a SEM, a scanning electron micrograph of neurons, and the neurons are what function in your brain to help your brain keep going. Okay, so these microscopes are pretty cool to be able to see those structures. Now there's two different kinds of cells that are out there. We already know that there's plant cells and animal cells, but they actually come in a variety of shapes and sizes. They do not all look the same. However, all cells are surrounded by a barrier called a cell membrane. And at some point, they do contain DNA. And if you are a fan of any sort of law show, or you just kind of know this just from talking to other people, DNA are what contain, that's our genetic material. So all cells, no matter what kind of cell it is, surrounded by a cell membrane and has some sort of DNA. Cells are classified into these two categories, like I said, and they are classified depending on whether they contain a nucleus. Now, a nucleus is a large membrane-enclosed structure that contains the cell's genetic material in the form of DNA. The nucleus controls many of the cell's activities, so it's kind of like the power brain center of the cell. All right, so the first type of cell that we're going to be talking about is a eukaryote, and these are cells that contain a nuclei. A nuclei is just a single nucleus. And then a prokaryote is a cell that does not contain a nuclei. So those are the two main differences between a eukaryote and a prokaryote, is that if it does have a nucleus, it's in a eukaryote. If it does not have a nucleus, it is a prokaryote. Okay, so what are some characteristics of these prokaryotes and eukaryotes? Well, the prokaryote. These cells have genetic material that is not contained in a nucleus. It just kind of free floats throughout the cell. Prokaryotes do not have a membrane-bound organelles. They are generally smaller and simpler than eukaryotic cells. And bacteria are considered prokaryotes because of the structure and how they are made up. Then eukaryotes. Remember, this is one that has a nucleus in which the genetic material is separated from the rest of their cell. And eukaryotic cells are generally larger and more complex than prokaryotic cells. They generally contain dozens of structures and internal membranes, so lots of different little organelles that get to float around in there. Many eukaryotic cells are highly specialized. That means they serve a specific purpose for that cell. It is not just an animal cell or just a plant cell, but it has an actual specific purpose throughout that organism. Plants, animals, fungi, and protists are all eukaryotes, whereas we said before, bacteria are going to be the prokaryote. Okay, that's it for this chapter section. So let's go ahead and do a little learning objective review for what we covered today. So remember, I will not be showing the answers to these on the screen, so you will either have to listen to what I'm providing you with the information, or you will have to go back and get it from the notes on your own. So it's up to you how you decide to do that. Okay, first learning objective review is, what is the cell theory? Well, remember, the cell theory states that all living things are composed of cells. Cells are the basic units and structure and function of living things, and new cells are produced from existing cells. Those three things, those define the cell theory. The next learning objective review is, how do researchers explore the living cell? Well, by using the various types of microscopes is how they will explore those cells, because cells are extremely tiny. The last learning objective review for this section, what is the difference between eukaryote and prokaryote? Well, the difference is that a eukaryote is going to have a nucleus, And a prokaryote, the nucleus is not contained, so they do not have a nucleus. Okay, that is it for this section of notes. Feel free to rewind, rewatch, pause the video to get everything that you don't have already, and make sure you save these pages, and we will put them in your interactive science notebook the next time we are in class. Thanks. See you later.